Hello and welcome to Research Matters podcast. Today Aishwarya Vishwamitra, will give you a summary of science stories that were published during the week on Research Matters. In your weekly dose of Indian science news today, I cover stories on diseases, drug treatments, and nanoforms of carbon. Have you heard of the neurodegenerative disease called Alzheimer's? Alzheimer's is a disease that causes the neurons in the brain to degenerate and die. In the brains of Alzheimer's patients, there are characteristic aggregates of a protein called tau. However, tau, being necessary for stabilizing the microtubules in cells, is found in everyone's brain. So why then are these aggregates formed? To answer that, it is essential to understand how the tau protein is found in the brain under normal circumstances, as liquid droplets. Researchers at the Indian Institute of Science, Education and Research, Mohali, characterized these droplets and discovered the underlying chemical mechanism behind the droplets' formation. Haven't we all seen what happens when oil is added to water? It collects into dense droplets and floats on top of the water. Similarly, a homogeneously mixed protein solution may demix via a process known as liquid-liquid phase separation into a dense phase comprising the liquid droplets and a light dilute phase. This happens naturally in all cells. Tau undergoes phase separation to form liquid droplets under natural conditions. The researchers found that, just like a coiled up spaghetti unit cannot interact with another coiled up spaghetti unit, tau cannot interact with other tau units either. But because of this phase separation, the tau proteins spread out and unravel, allowing them to interact with each other. This is how it forms its dynamic fluid state. Now that we know how it exists naturally, let's understand the aggregation. This fluid state is precarious. Specific mutations or other factors can convert these transient interactions into permanent, persistent ones, causing the proteins to solidify into ordered aggregates, disrupting the functioning of the neurons. The researchers are now trying to understand this process better, find out what causes these transformations, and see if it can be prevented. Continuing with the ongoings of the brain, we all know that the brain is made up of cells called neurons. These neurons form intricate pathways within the brain, enabling you to do a host of activities including seeing, making decisions, and remembering. However, these pathways are so interconnected that it is difficult to pick out a single pathway to understand its function in the brain. To combat this problem, a technique called chemogenetics uses designer receptors to understand how neurons are activated. How do the designer receptors work? They interact with the inert chemical clozapine N oxide or CNO in the brain and causes an increase in the calcium ions within the cell. This leads to the release of chemical messengers called neurotransmitters, which activate the next neuron connected, then the next, and so on, activating all the neurons in that particular circuit and singling it out. However, a study by researchers at the Tata Institute of Fundamental Research and Jawaharlal Nehru Center for Advanced Scientific Research found that this technique is unreliable because different concentration doses of CNO cause different effects on neuronal properties. They used 1 micromolar and 20 micromolar dosages to show this. Strengthening of connections between neurons helps in generating memory. The researchers observed that high doses of CNO prevented strengthening of connections, while low doses facilitated it. The different doses of CNO had nearly opposing effects. Thus, they concluded that since using different doses causes different changes, these designer receptors cannot be used as the technique isn't consistent. Let's move on to another disease, and this one requires a revamp in its treatment. Prostate cancer is one of the 3 most common cancers in men. It is caused when cells of the prostate gland start growing abnormally due to an elevated level of androgens. The prostate gland is a male reproductive organ, and androgens are hormones required for development and maintenance of the male reproductive system. I said elevated level of androgens causes this. Therefore, the common treatment is called androgen deprivation therapy, by which androgen production is reduced or the androgen receptors are blocked. But its drawback is that cancer eventually relapses or returns in a few years in many of the affected men when this therapy is used. It comes back more aggressive and becomes resistant to anti-androgen drugs. It then progresses into another aggressive cancer, neuroendocrine cancer. But don't worry, scientists have found that increased expression of a gene called SPINK1 is one of the factors involved in prostate cancer. In a study done by Indian Institute of Technology, Kanpur, in collaboration with CSIR, Lucknow, researchers have highlighted how androgen deprivation therapy aggregates prostate cancer instead of curing it, and have even suggested an alternative approach. Here's what they showed. The androgens send out signals that control SPINK1's gene expression. Elevated level of androgen receptors express lower levels of SPINK1 and vice versa. Thus, blocking the receptors altogether caused SPINK1 expression to go way up. When the tumors were implanted in mice, they found that giving androgen deprivation therapy caused a robust increase in SPINK1 expression and displayed markers for neuroendocrine cancer. Thus, the researchers realized that repressing the expression of SPINK1 gene was the way to go. They went on to find other proteins involved in the androgen signaling pathway, which represses SPINK1's expression. Considering that androgen deprivation therapy's use is widespread, action needs to be taken to ensure well-informed decisions are made while treating prostate cancer. While for some diseases treatments need to be revamped, other diseases are difficult to identify in the first place. Tuberculosis is one such disease. Mycobacterium tuberculosis is the bacterium that causes the infection and belongs to the family Mycobacteriaceae. This family contains different strains of bacteria which can cause other TB-like disease, which mimic tuberculosis in signs and symptoms but have a different clinical treatment. This obviously leads to confusion during diagnosis. Patients are started on the anti-tuberculosis treatment even if they don't have TB, but just a TB-like disease. Incorrect diagnosis leads to a waste of time and resources and puts the patient at risk. The solution? An international study, including researchers from India, have designed a rapid, cost-effective, and readily accessible triage test kit to detect the presence of active TB. So what is a triage test? A triage test aims to determine who should undergo the more extensive and expensive confirmatory diagnostic testing and who should not. Tested on patients with suggestive TB symptoms, the researchers measured the levels of different proteins in their blood. 4 host proteins, which stimulate immune responses, they are 3 types of interleukin and vascular endothelial growth factor, are found circulating in the blood of active TB patients. These samples were analyzed using a machine learning technique that identified these 4 proteins, and the algorithm was able to detect the presence of active TB with a high percentage of specificity and sensitivity, telling apart true positives and true negatives. They didn't stop there. The researchers then developed an ultra-sensitive immunoassay to improve the performance of their test model and screen protein levels in a small quantity of blood. Trials are going to start soon. Did you know that nanomaterials are used for drug delivery in the human body? Apart from medicine, they have a variety of uses, and there are so many nanoforms of carbon, nanotubes and nanocones, just to name a few. Well, add one more to that list. Researchers from the Indian Institute of Technology, Bombay, have designed nanocarbon florets, and just as the name suggests, they are shaped like marigold flowers. They aren't just pretty objects. Impurities stick to the surface of the floret when water passes over it, thus removing impurities from the water. Plus, they're reusable. All it takes is a wash with a mild acid and they're as good as new. With the added benefit of being stable over a wide temperature range, nanofluorides are a convenient and sustainable solution to decontaminate water. They remove the impurities using a process called adsorption, which means impurities stick to it. But what makes them better than other adsorbents? Unlike biomass-based adsorbents, synthetic adsorbents are reusable, but they are not biocompatible and are hence bad for the environment. Unlike other carbon-based materials, these nanofluorides are hydrophilic, meaning water sticks to it. The fluorides have a large accessible area for adsorption and are chemically stable in highly acidic or alkaline water. The researchers have applied for a patent. Thank you for listening to this episode of Research Matters podcast. See you next weekend. To hear more such podcasts and news, please log on to www.researchmatters.in.